When you look at a plate of food and think about the concept of diversity on your plate, you probably think about the different types of food you are eating. Here we have kokoda, the national dish of Fiji, which includes a variety of ingredients like coconut milk, fish, tomatoes, peppers, onion, lime, and cilantro. However, this is just one way to think about the diversity on your plate. On a smaller level, the ingredients we recognize in our food are all made up of an assortment of molecules. Take our tomato and the kokoda as an example. A variety of molecules make our tomato a tomato. Some of these, like macronutrients including carbohydrates, fats, and proteins, are molecules that are essential to our diet and are often displayed on nutrition labels and in food composition databases. Similarly, micronutrients, like minerals and vitamins, are essential components of our diet that we consume at lower quantities. But our food is more chemically complex than just these macro and micronutrients. Before tomatoes cross our plates, they are plants that are exposed to a specific growing environment that can include adverse weather conditions or insects and pests. How do plants survive these stressors? The answer is specialized metabolites, which are small molecules that perform essential functions to communicate changes in the environment, protect the plant, and facilitate the development of the plants we eat and harvest. These small molecules are also associated with some of the unique properties we associate with foods, such as certain colors and tastes. Have you ever bitten into a pepper and felt an immediate burn? That burn happens to be because of a small molecule called capsaicin. But small molecules do more than just contribute to food's unique sensory properties. There has also been evidence that small molecules in food can have health benefits too. The small molecules we've described are all things we can describe as endogenous, or molecules that are made by the plants themselves. However, this might not be the entire picture of what is in our food. The system of how a plant is grown, harvested, shipped, and stored can introduce food compounds like pesticides, herbicides, fertilizers, preservatives, and additives to our food. We call these compounds exogenous molecules. The PTFI is interested in identifying the diversity of endogenous and exogenous small molecules in the foods that people eat around the world. To do this, we use an untargeted metabolomics approach that is based on liquid chromatography coupled with mass spectrometry, which enables us to discover and identify endogenous and exogenous small molecules in our food. In this module, you will be provided with an overview of the standardized untargeted metabolomics protocol developed by the PTFI to analyze a small molecule fingerprint of different foods. You will also observe the PTFI Metabolomics method in action and learn about potential research applications of these analytical techniques. Let's get started.